Day everyone. Now we will be talking about cells. So overview, um since the late 1800s, cell research has been exceptionally fruitful, no, and it has provided us with four concepts collectively known as the cell theory. So a cell is the basic structural and functional unit of living organisms. When you define cell properties, you define the properties of life. The activity of an organism depends on both the individual and the collective activities of its cells. And according to the principle of complementarity, the biochemical activities of cells are dictated by the relative number of their specific subcellular structure, and continuity of life has a cellular basis. So we are going to begin the idea that the cell is the smallest living unit. No, cell is the microscopic package that contains all the parts necessary to survive in an ever-changing world. So again, um it follows then pag may disturbance dun sa homeostasis ng cell, it will eventually lead to disease.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Okay, so always remember, a human cell will have three main parts. That's your plasma membrane, the outer boundary of the cell, the cytoplasm, the intracellular fluid packed with organelles. And those organelles, no, these are small structures that perform specific cell functions. And then you have your nucleus. This is an organelle that controls your central cellular activities. So if we chemically analyze the cells, we find that the cells are composed chiefly of your carbon, your hydrogen, your nitrogen, and your oxygen, and trace amounts of several other elements. So anatomy of the cell again. First is we have your plasma membrane, the outer boundary of the cell. Your cytoplasm, which is an intracellular fluid packed with organelles, and these organelles, these are small structures that perform specific cell functions. Then your nucleus, this is an organelle that controls your cellular activities and lies near the cell center. So the nucleus, no, anything that works best when it is controlled. So for cells, the control center is the gene-containing nucleus. So the nucleus can be compared to a computer, no? Merong construction boss, merong board of directors, all rolled into one. So it houses your DNA, which is a genetic material much like a blueprint that contains all the instructions needed for building the whole body, or more specifically the proteins. So the nucleus, no, it has also parts. So first is your nuclear membrane or the nuclear envelope, your nucleoli, and chromatin. You also have your nuclear pores. So your nucleus or your nucleus, it's bounded by a nuclear envelope or nuclear membrane. This is a double membrane barrier separated by a fluid filled space. So the nuclear membrane, they encloses a jelly fluid like nucleoplasm in which other nuclear elements are suspended. So at various points, no, there will be the nuclear envelope is punctuated by your nuclear pores. So your nuclear pore complex is an intricate complex of proteins, and they line each pore, forming an aqueous transport channel and regulating entry and exit of molecules and large particles into and out of the nucleus. Then your nucleoli, also known as your little nucleus, these are the dark staining spherical bodies found within the nucleus where ribosomal subunits are assembled. So most ribosomes eventually they migrate into the cytoplasm where they serve as the actual sites of protein synthesis. Typically, there are one or two nuclei per nucleus, but there may be more. So when a cell is not dividing, the DNA is combined with protein and forms loose networks of bumpy threads called chromatin, and that is scattered through the nucleus. So when a cell is dividing to form two daughter cells, the chromatin thread cells coil and condense to form dense like rod-like bodies called chromosomes. So your plasma membrane. Your plasma membrane is a fragile transparent barrier that contains the cell contents and separates them from the surrounding environment. So we have here the fluid mosaic model. No, it just shows the structure of the plasma membrane, in wherein there is a two lipid layers arranged tail to tail in which the protein molecules float. So you have your polar heads, which is hydrophilic, meaning water loving. Don't get confused. Polar head, water, those are hydrophilic, which means water loving, and they're the one that is attracted to water. So andyan sila both sa inner and outer surface of the membrane. While your non-polar tails, these are hydrophobic, they hate water. So ang mga hydrophobic, nilalagay natin sila sa center of the membrane. So dahil dun sila sa center of the membrane, no, it makes the membrane impermeable to most water soluble molecules. So, di ba, they can filter out. So, you also have your cholesterol in there. This helps to keep the membrane fluid. You also have your proteins, which is sila yung responsible talaga for most of the specialized function of the membranes. Like, later on, you will know what are these proteins for receptors, for binding sites, for transporting enzymes, mga ganun. Next is you have your glycoproteins. These are branching sugar groups. Yan. Attached to most of the proteins, no, abutting the extracellular phase. You also have your glycocalyx, a fuzzy, sticky, sugar-rich area cell surface. So, you also have your tight junctions. These are impermeable junctions that bind cells together into leak-proof sheets that prevent substances from passing through the extracellular spaces between cells. So, example, in your small intestine, these junctions prevent nila yung digestive enzymes na pumasok sa bloodstream. Your desmosomes, no, these are anchoring junctions scattered like rivets along sides of abutting cells. So, para silang glue, they prevent the cells to mechanically stress from being pulled apart. So gap junctions, these are communicating junction, no? It self-explanatory. It's mainly to allow communication. So neighboring cells are connected by hollow cylinders composed of proteins called your connexons that span the entire width of the abutting membranes. Next is your cytoplasm. Your cytoplasm is a cellular material between the plasma membrane and your nucleus. So this is the site of the most of cellular activities and also called the factory area of the cell. So your cytoplasm, we have your post of your cytosol, ito yung fluid na doon nag-float ang mga cytoplasmic elements. No? Dissolved in that cytosol is just largely water, and there are nutrients and a variety of other solutes. Then, yung mga nakaspend doon sa cytosol na yon, fluid, is your organelles. Ito yung makina talaga ng cell natin. So, each type of organelles, they have a specific function to make the cell function as a whole. So, may mga inclusions naman ng cell, yung mga chemical substances that may or may not be present depending on the cell type, like your lipid droplets that can be found in fat cells, or sa liver mo, may mga glycogen granules sa cells mo, or sa skin mo, di ba yung mga pigments such as melanin. Then your little organs, these are specialized cellular compartments or structure, each performing its own job to maintain the life of the cell. So this is the cytoplasmic organelles. Next is your mitochondria. Syempre, alam naman natin sino si mitochondria. This is a tiny sausage-shaped organelles, but in living cells, they squirm, elongate, and change shape almost continuously. So meron din siyang outer membrane. Yung outer membrane niya is smooth and featureless, pero yung inner membrane niya has shelf protrusions called the cristae, your cristae. So this is the powerhouse of your cell, and the mitochondria supply most of this ATP. So your ribosomes, there are tiny bilobe dark bodies made of proteins and one variety of RNA called your ribosomal RNA. So your ribosomes, ang lagi niyong tatandaan sa ribosomes is this is the site of the protein synthesis in the cell. So some of these ribosomes, they float free, while others attach sila sa membrane. So yung mga naka-attach sa membrane, that forms your complex na tinatawag natin rough endoplasmic reticulum. So your endoplasmic reticulum, we also call this the network within the cytoplasm. And it's a system of fluid-filled cisternates that coil and twist through the cytoplasm. And this serves as a mini circulatory system for the cell. So there are two forms of your rough ER. So rough ER, these are studied with ribosomes. Kaya tinatawag na rough. While your smooth endoplasm, tingnan nyo, merong mga ribosomes. Yung smooth endoplasmic reticulum, continuous siya pa rin sa rough ER. And ang main function nito is for lipid metabolism. So, cholesterol and fat synthesis and breakdown and detoxification of drugs and pesticides. Next is your Golgi apparatus. Your Golgi apparatus, they appear as stacks of flattened membranous sacs associated with swarms of tiny vesicles. So, this is the principal traffic director for cellular proteins. And ang trabaho nila is to modify and package the proteins na pinadala ng rough ER via transport vesicles. Yan. So, vesicles containing proteins destined for export. Yan. Pinches of other vesicles containing lipids designed for the plasma membrane and packages digestive enzymes into membranous lysosomes that retain in the cell. So, your lysosomes, we also have your lysosomes, which is we call the breakdown bodies. These are membranous bags containing mga powerful digestive enzymes, lysosomal enzymes, and sila yung capable na nagdadigest ng mga walay pulos na cell structure and most foreign substances na pumapasok sa cell. Next is your peroxisomes. So peroxisomes are membranous sacs containing powerful enzymes pa rin. And this is important for oxidase and catalase. So, Your oxidase, they use molecular oxygen to detoxify harmful substances, including your alcohol and formaldehyde. Next is your catalases. They convert your hydrogen peroxide to water. Next is your cytoskeleton. Your cytoskeleton is an elaborate network of protein structure that extends to the cytoplasm, and it acts as the cell's bone and muscles. No? Sila yung nagpro-provide ng internal framework, no, na determines the cell shape, supports other organelles, and provide the machinery for intracellular transport and various type of cellular movements. So your microfilaments, these are most involved in cell motility in producing changes in cell shape. So your microfilaments, these are involved in the cell motility and nagpo-produce ng changes sa cell shape, while your intermediate filaments, these are strong na parang rope-like, and sila yung nagpo-form ng desmosomes. And your microtubules naman, tube-like sila. So your intermediate filaments are rope-like. Your microfilaments made for motility. And your microtubules naman is parang tubo. So centrioles, these are rod-shaped bodies that lie at right angles to each other. So they are made of a pinwheel array of the microtubules, and their role is to generate microtubules entering cell division. So the centrioles, these are the one directing the formation of your mitotic spindle. So we also have your cilia and your flagella. These are whip-like cellular extensions that move substances along the cell surfaces. No, ang plus nila is mag-move ng something. And flagella, it's longer than the cilia. So yung cilia, sila yung nag-transfer ng something, while yung flagellum naman, they are transferring the cell itself. So microvilli, so these are tiny, pag buzzword niyan is finger-like extensions na projected from the exposed cell surface. So cell diversity, so our cells are very diverse. They have different purposes and functions. So you have cells that connect your body parts like your fibroblasts and erythrocytes, and you have also cells that function to cover and line your body organs. There are also cells that move organs and muddy parts like your skeletal muscle cell and smooth muscle cell. There are cells that store nutrients like your fat cells. There are cells that fight diseases like your macrophage. And there are cells that gathers information and controls body function like your nerve cell. And there are also cells that their purpose is for reproduction. So let's proceed to cell physiology. Okay, let's watch this first.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose, but what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We would still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically is going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates, and when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside. And this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf, and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit, they sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. All cells have membranes, but not all cells have a wall, but plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Ayan, so it is important that you really understand solutions before we dive into an explanation of membrane transport. So ayan, so a solution that's a homogeneous mixture of two or more components. So a solvent that is a substance present in the largest amount, yung pinakamalaki dun sa solution. While your solute, these are components or substances present in smaller amounts. So together, that would make your solution, which is a homogeneous mixture of two or more components. We have your intracellular fluid. So intracellular fluid meaning yung fluid na nasa inside ng cell. So collectively, the nucleoplasm and cytosol. So this solution contains small amount of gases, your oxygen, carbon dioxide, nutrients, and salts dissolved in water. Next is your interstitial fluid. So this interstitial fluid is the fluid that continually bathes the exterior of the cells. Kita niyo nasa exterior siya. And this contains nutrients, regulatory substances such as your hormones and neurotransmitters, salts, and waste products. So we have what we call the selective permeability. So meaning some molecules can cross the membranes while the others cannot. No? It is designed so it can allow nutrients to enter the cell, but it can keep many undesirable substances out. So we have the types of membrane transport. So first is we have passive process. So passive lang siya. So without any energy, no? Kasi yung substances, nagmo-move sila sa yung plasma membrane in two ways. Pwede siya passive and pwede siya active. So sa passive muna tayo. So sa passives, kasi sinabi siyang passive, di ba, without energy. While your active process naman, it needs energy para ma-transport niya yung tinatransport niya. So passive process, walang energy, doesn't need energy to transport. Well, yung active process mo, it needs energy. So diffusion. Diffusion is the process by which molecules move away from a region where they are more concentrated to a region where they are less concentrated. So the overall effect of this erratic movement would be that the molecules move down their concentration gradient. So yun, the greater yung difference ng concentration ng between the two areas, mas dadali yung pagdi-diffuse niya. So ayan, we have your simple diffusion. Okay, so your simple diffusion solutes are transported this way as either lipid soluble, no, and or pinapass through lang sila through membrane pores. So directly lang, simple diffusion of fat-soluble molecules directly through the phospholipid layer. While your carrier-mediated protein, your carrier-mediated facilitated diffusion, no, because meron na siyang carrier, that's via a protein carrier specific for one chemical. And the binding of substrate causes transport protein to change shape. So, meron din tayong tinatawag na channel-mediated facilitated diffusion. So, through a channel, di ba? Through a channel. So, itong channel ito is protein pa rin. And mostly nag-utilize ng ganitong system is yung ions. And they are selected on the basis of their sizes and charge. Next is your osmosis. So, diffusion of solvents such as water through a specific channel protein. That is your aquaporins, no? Or through the lipid bilayer. So, yan. Filtration. Filtration is the process by which water and solutes are forced through a membrane by fluid or hydrostatic pressure. So, the gradient is a pressure gradient na actually pushes the solute containing filtrate from the higher pressure area to the lower pressure area. So, next is we have your exocytosis. So, out of the cell, it moves substances out of the cell. The product to be released is first packaged into small membranous sac called a vesicle. Then it will migrate to the plasma membrane, fuses with it, and ruptures, spilling the sac contents out of the cell. Endocytosis means into the cell, so process that takes up or engulf extracellular substances by enclosing them in a small membranous vesicle. So, once the vesicle or sac is formed, it detaches from the plasma membrane and moves into the cytoplasm, where it typically fuses with a lysosome and its contents are digested. So, we have your phagocytosis. So, phagocytosis is the cell engulfs a large particle by forming projecting pseudopods around it and enclosing it with a membrane sac called a phagosome. So, ayan, once a phagosome is combined with your lysosome, the undigested contents remain in the vesicle, now called a residual body, or are ejected by exocytosis. Next is your pinocytosis. So, when you say pinocytosis, the cell gulps a drop of extracellular fluid containing solutes into tiny vesicles. So, it's more of like a cell drinking. So, receptor-mediated endocytosis, extracellular substance bind to specific receptors. So, self-explanatory, may receptor proteins, enabling the cell to ingest and concentrate specific substances. Ayan. So, cell-eating phagocytosis. Diba? Phagocytosis. Cell-drinking pinocytosis. And yung receptor-mediated endocytosis by specific target molecules. Okay, next is cell division.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes, very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends, kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Ayan, so always remember that the cycle has 2 major periods. So first is your interphase, that's in green, in which the cell grows and carries on its usual activities. While the actual cell division or the mitotic phase, that's in yellow, during which it divides into 2 cells or reproduces itself. So in your interphase, your G1, that is when the cell is metabolically active, synthesizing proteins rapidly and growing vigorously. While your S phase naman, the DNA is replicated, ensuring that the 2 future cells being created will receive identical copies of the genetic material. Your G2 naman is where the enzymes and other proteins are needed for division and are synthesized and moved to their proper sites. By then, the cell is now ready to replicate. To divide, I mean. So your DNA. So your DNA is essentially a double helix. No, para siyang ladder-like molecule that is called into a spiral staircase shape. So once the DNA helix is uncoiled and the hydrogen bonds between its base pairs are broken, each nucleotide strand of the DNA acts as a template for constructing a complementary strand. So DNA polymerases work in one direction only. So the 2 new strands, your leading and your lagging strands, are synthesized in opposite directions. So one strand, the leading strand, is synthesized continuously once primed, following the movement of the replication fork. Then the other strand, called the lagging strand, is constructed in segments in the opposite direction and requires the primer initiate replication of each segment. So, yan, your cell divisions, that's mitosis, your division, no? In the division of the nucleus, and cytokinesis is the division of the cytoplasm. So, in your mitosis, you have your PMAT, no? PMAT, prophase, metaphase, anaphase, and telophase. So, ayan, you have seen the video. So, let's just do a quick run through. So, during the early prophase, the chromatin condenses, forming bar-like chromosomes. Then, each duplicated chromosomes consist of 2 identical threads called your sister chromatids. Then, they are held together at the centromere. So, the centromeres acts as a focal points for the growth of a microtubule assembly called the mitotic spindle. And as the microtubules lengthen, they propel the centrosomes toward end of the cells. So microtubule arrays called asters or stars extend from the centrosome matrix. Your late prophase, the nuclear envelope breaks up, allowing the spindle to interact with the chromosomes, and some of the growing spindle microtubules attach to kinetochores as special protein structures at each chromosome centromere. So such microtubules are called kinetochore microtubules. Then the remaining spindle microtubules are called polar microtubules, and the microtubules slide past each other, forcing the poles apart. So the kinetochore microtubules pull on each chromosome from both poles in a tug of war that ultimately draws the chromosomes to the center or equator of the cell. So actually, that this PMAT is best learned through a visual yung video kanina. Or you look for more videos that will help you understand. Okay? So, protein synthesis. Your DNA, that's the master blueprint for protein synthesis. So, it's like, it's like a strip of magnetic recording tape, no? So, ayan, your RNA naman, your RNA is a second type of nucleic acid that it decodes and is a messenger for task of specifying yung structure ng proteins to be built at the ribosomes, So, ayan, you have your transfer RNA, your ribosomal RNA, and your messenger RNA. Yan. So, protein synthesis. First is you have your transcription that involves the transfer of information from DNA's base sequence into the complementary base sequence of mRNA. So, each 3-base sequence specifying a particular amino acid on DNA gene is called a triplet, and the corresponding 3-base sequences on mRNA are called codons. So, translation, that's the information carried in mRNA molecules, is decoded and used to assemble proteins, and this occurs in the cytoplasm. So, thank you very much.